Good morning to all. It is with great pleasure that I extend this invitation to you for ICPTM 3.0. Your presence would add a touch of warmth and joy of gathering. We sincerely hope you can join us for an unforgettable experience. Please be seated, everyone. So, I would like to invite Mrs. Ranu Sharma ma'am to welcome Dr. Kamal Sharma ma'am, Professor BN IPS Udaipur. Ma'am, please. तालियां बजेगी तो बड़ा मजा आएगा. Thank you so much, ma'am. Now, I would like to invite to welcome Dr. Shikha Agarwal ma'am. So, I invite to our sir, please welcome Dr. Shikha Agarwal ma'am, Department of Chemistry, MLSU. तालियां बजती रहनी चाहिए. Thank you so much, ma'am and sir. Now, I would like to invite Mr. Dilip Kumar sir to welcome Dr. Priyam Vadasik ma'am, Miranda House, Delhi University. Thank you so much, ma'am and sir. Now we will start our technical session for oral presentation. So I would like to invite Dr. Rajesh Khaturiya sir to continue and announce the list for this session. Sir, please. It's over to you, sir.
Thank you, Dr. Gunjan Janol ma'am. Good morning to all of you. Let's start this technical session for this. First of all, I will invite the moderator of this session, Dr. Kamal Sharma, Dr. Shikha Agarwal, and Dr. Nirmala Singh. I request all the presentators to please limit their presentation within 8 minutes. First bell ring will bell after 6 minutes, and next bell will ring after the 2 minutes from the first bell. The rest of the presentation is total is 8 minutes. So now I request for oral presentation. First presentation is OP 32.
जिसकी पीपीटी यहाँ सबमिट नहीं हुई है तो वो प्लीज सेव करवा दें. OP 18 19, 28, 29, 30, 31, 32, 33 and 34. Remaining कोई भी है तो वो प्लीज यहाँ पे सेव करवा दें. 20, 21, 22, 22, 23, 24, 25, 26 and 27.
From natural marine extracts and approach for development of new antigens. So the world of malaria is facing an imminent threat, which is resistance, be it insecticide resistance or anti-malarial resistance.
Hello.
So the world of malaria is facing an imminent threat. So the current frontline drugs, which are artemisinin derivatives, so they are facing resistance. So this means there's a need for novel molecules, and then natural products have always been a source for these new drugs. Even today, most of the most of the drugs in the market are coming from nature only, either modified or semi-modified. So in previous lectures also, yesterday and day before, so we saw that 70% of the drug discovery, they come from natural sources. And antimalarials, which are frontline drugs, artemisinin and chloroquine, they were also isolated from plant and tree species. So my work plan included that I tried to reach, so the whole idea was that India is surrounded by 3 marine bodies. So we tried to reach out to the different scientists across the coastal line of India so that they can provide us with some marine extracts. Because since we have 3 different bodies, so we were expecting that we would get a lot of diversity in the extracts, a lot of chemical space and a lot of diversity. So there will be a lot of different secondary metabolites as compared to the terrestrial ones. And it remains unexplored. The Indian coastal region, it remains unexplored. So we try to do that. So this is the methodology. So basically we screen them for any antimalarial insecticide activity. And so these were the results. So actually the first batch that we received were from CSIR NIO, which is in Goa. So they gave us around 50-60 marine bacterial extracts. So but they did not show any anti-plasmodial activity. So in vitro, there was no activity against P. falciparum, which is the positive agent of malaria. You can see that here in the microscopic slides also. There's the same morphology in the control ones and the treated ones, and the chloroquine is the positive control. But when we checked for the insecticidal activity, so we saw that these marine bacterial extracts are killing Anopheles larvae very potently. So these graphs represent all these marine extracts showing 80 to 100% motility at 500 and 125 ppm, and even at lower concentrations they were showing 80 to 100% motility. many of them. So we checked for more activities, like there are different stages in the in the development of the mosquito. So we checked at the larvae and we checked at the egg level also. So these two images, these two graphs are showing the ovicidal activity. So this basically is showing the transformation of the eggs to larvae. So the eggs hatch and the larvae comes out. So here we see that the percentage, so you can see this compound 740. The reduction of these larvae formation is, you can see the graph is very low. So there will a very low number of larvae are coming from those eggs. So this compound is causing even reduction of larvae formation. So it has it is acting on two stages, larvae also and eggs also. And this C graph is actually we checked on another mosquito which is dengue vector, Aedes aegypti. So we checked it is acting on other mosquito species also. So this we checked 740 and 707. It also showed activity against Aedes aegypti also. So some of the extracts had specific activity for malaria vector, and two of these extracts also had activity against dengue vector also. And this graph D, this graph is representing, actually we checked in semi-field settings. So what we did was this highly prone area of mosquito development we see in Mewat, Haryana. So that is an area where we see a lot of larvae formation, and there is a lot of incidence of malaria there. So we checked for activity in the field also. So if it is acting, the extracts are acting not just in the lab and the lab strains, but also in the field. So even in the field, this one extract 706, it had 80 to 100, it showed 80 to 100, almost 100% mortality at 125 ppm. So, and this graph is representing E1 is adulticidal activity. So we checked at adult stage also. So there was no activity at adult stage. There was activity at larvae in Aedes aegypti in field settings also, and we also saw egg formation, and the larvae formation was reduced. So this is the phylogenetic analysis of the hits where we received from. So you can see we received from Lakshadweep, Ratnagiri, local, many different parts of Indian coastal region. And the second batch that we received was 70 invertebrate extracts we received. So before going to any other activity, we checked for antiplasmodial activity with the cytogreen assay. And this heat map is showing the activity of the 70 extracts. And the bar that you see, which is dark in color, so this is chloroquine. So the more the band is dark, so the more activity it has. So you can see this band is even better than chloroquine. So all these bands which are anything which is comparable to chloroquine is a good hit. So we in the first screening we received these extracts and we are planning to investigate them further. And this is the microscopic view of it. So conclusion is that we are out of 200 marine extracts. The first batch bacterial extract was in vitro, it did not have any in vitro activity against Plasmodium, but larvicidal activity it had, and like I said, 100% motility at 125 ppm, many extracts were showing, and 740 reduced even the X species, and out of the 70 marine extracts, the 7 extract has potential hit. So we are planning to investigate them further so that we can take them as hits to leads for any drug development. And we are also planning the larvicidal bacterial extract to make them as a working formulation in field settings. So it can be also translated to field settings. So these are the accomplishments. My PI and the collaborator Dr. Nursing and ICMR and CSR for my funding and ACSR for my these are my accomplishments. Thank you so much.
a drug because you are working in the direction where they can act as a drug?
Actually, in anti-malarials, many bacterial extracts their metabolites, secondary metabolites, alkaloids. These are have shown in many reports have shown very good anti-malarial activity. Yeah, secondary metabolites from the bacterial extracts. So basically, these are also secondary metabolites only. So alkaloids and so.
Thank you, Cherish. Now I request to OP18 to present their oral presentation.
This is OP?
OP13.
Good morning, Namaste. Myself, Bharadwaj, and I am from Hasras College, University of Delhi. And today I will be presenting my work, an easy and convenient protocol for specific synthesis of gaminophin drug and its derivatives. Gaminophin drug is well known for its muscle relaxation activity, and it is often given with other medications like paracetamol to relieve pain and fever at the same time. And it has worldwide distribution too. So previously synthesized method is there are so many companies, they are selling their gaminophin by using this particular methodology. They are using this alpha amino ester as a starting material. They are reacting, their reaction condition is DMSO and T2C3 as a base, reflex, and 10 hours of reaction time, which is giving this product. This base is actually abstracting the proton from this NH2 moiety. This is simple substitution reaction, but we must know that to prepare this moiety it's very difficult, and this is not easily available in the market, and if it is available in market, it has a very high cost. Based on these advantages like high reaction time, non-functional no functional group tolerance, mixture of product, they are getting a mixture of product like 52% of camilophin, because the camilophin R isomer works very well, but S isomer does not work for relaxation of pain. So based to overcome from these advantages, we have approached new synthetic methodologies. We are using very simple and economical chemicals, starting from mandelic acid, and we have very nice diversification in isomers too. And we are using mandelic acid, R-mandelic acid, and S-mandelic acid. When we are using R-mandelic acid, we are getting almost more than 90% are camilophin. And when we are using L-mandelic acid, we are getting L-camilophin. So this is very simple chemistry. We have discovered the catalytic amount of this boric acid activates the carbonyl center of mandelic acid, which gives this alpha hydroxy ester product. Further, in the presence of SOCl2 as a solvent and as a catalyst, we are getting this chlorinated product for the further reaction. And the chlorinated this chlorinated product, when reacted with different type of amines, we are getting desired product in very nice yield, excellent. So there are the very nice advantages of our this synthetic morphology, which is functional group tolerance and let us say synthetic group, high yield of product, and very less time is required. We are only using 4 hours for last compound synthesis. I mean, starting from this to get this product, we are using only 4 hours. And stereo-specific product and diversification in the derivatives, we have synthesized it, this cavalopin in gram scale, also we have synthesized in 20 gram. And it's a very nice economical method. And this is the table for optimization. Final product we are getting in 1 hour, and that is 92% of yield. These are the few substrate scopes for our methodologies. I cannot show all the synthesized compound here. And these are secondary amines and these are primary amines. We have synthesized more than 40 compounds over here. Further, after the synthesizing and the characterization done, we go for the biological studies, and in starting we check the cytotoxicity of these compounds against Hep-G2 cells, and among these compounds, 3 compounds, LTC 7103, 7107 and 7116 shown no toxicity, and other has toxicity level less than 15 micromolar. And further, we checked IC50 values against Plasmodium burgii at liver state, and we found that these 4 compounds, LTC 7107, 7163, 7164 and 7169 are very nice and having IC50 values less than 10 micromolar, and our hit compound was 7107 having 1.63 IC50 value. As the conclusion of my presentation, stereospecific synthesis of more than 40 compounds, functional group tolerance, latest synthesis group, high yield products, less time required, economical route, diversification in derivatives, and we have done in vitro evaluation of compounds in liver stage. Further, we are planning for to perform anti-malarial acids. I would like to thank you, my supervisor, Dr. Vijay Srati sir, Hansraj College, University of Delhi, and UC, Delhi University Science Instrumentation Center, and Pacific University for giving us chance, and funding agencies like DICS, Department of Science and Technology. Thank you, sir.
Nice presentation. I would like to ask, as you told that aminosin is already FDA approved, for which clinical condition has been approved?
But it is in use.
It is in use for what condition?
Conditions?
Which disease?
It is simply used for the muscle pain relaxation. And sometimes cramps, जो cramps होते हैं उनके लिए cramp के लिए भी यूज होता है, abdominal pain के लिए actually यूज किया जाता है.
And how come it is better than the other drugs available in?
No, actually it is not, ये ना मतलब अकेले नहीं दिया जा रहा है, ये उसके पैरासिटामोल के साथ दिया जाता है, additive.
But there are other drugs also which are already combined with paracetamol and other non-steroidal anti-inflammatory drugs. How come aminosin is going to be better than the available drugs?
Actually, we are trying to find the activity of this amylopin and its derivative against malaria parasite. Yes. Actually, ये काम भी चल रहा है, ये पब्लिश नहीं हो रहा है.
2019.
Good morning to one and all present here. I, Dr. Assistant Professor, Department of Chemistry, Bahir University. My title of presentation is Evaluation of Dietary Status of Adolescent School-Going Boys of Himachal Pradesh. Given the paucity of data on the nutritional standard of the Himachali children, the present study was conducted to assess the physical growth and malnutrition of adolescent school-going boys of Himachal Pradesh within the age of 15 to 17 years. A cross-sectional study was adopted in the present study, which was conducted in the multi-district of Himachal Pradesh. To avail the logistic advantage, a wider range of geographical location, including hilly region of the districts were excluded. Only non-hilly zones and valley regions of the district were selected. This includes 3 subdivisions of Mandi district, Mandi Sadar, Bal and Sundar. 298 boys were selected from both government and private school of the non-hilly region of the Mandi district of Himachal Pradesh. Height, weight and body mass index were determined. Strength was evaluated from height for age Z-score and thinness, overweight and obesity were estimated from BMI for Z-score using WHO recommendation. The mean height varied between 167 to 168 centimeter. Weight varied between 52 to 56 kilogram and body mass index between 18.7 to 19.7 kilogram per meter square. The overall prevalence of stunting and fasting were found in 5.7 and 15.8% respectively. The overall prevalence of overweight and obesity was 5.4% and 1.7% respectively. The table 1 indicates the height increases shows with increasing age group. It shows height increases with age. At the age of 15 to 16 years, the mean height increases the government school boys as compared to private school boys. But the statistical difference was not found to be significantly. At the age of 17 years, the private school boys were taller as compared to the government boys, and the difference was found to be statistically significant. Table 2. Table 2 indicates it shows increasing trend with weight. At the age of 15, 16 years, the mean weight increases, the government school boys were increases as compared to private school boys. But the mean difference was not found to be were found to be statistically significant, while at the age of 17 years, the private school boys were heavier as compared to government school boys. But the mean difference was not found to be statistically significant. The table 3 shows body mass index. It also shows increasing trend with age. The body mass index at the age of 15, 16 and 17 years increases with age, but the mean difference was not found to be statistically significant. Despite the category of school, the government school boys were have heavier body mass index as compared to private school boys. The table 4 indicates the prevalence of stunting among Himachali boys. The prevalence of stunting was found to be 15.8%, and none of the boys were found to be severely stunting. The figure 1 indicates the different categories. There are 5 different categories of malnutrition: normal, thinness, over-thinness, overweight and obesity. These are the 5 categories of malnutrition. We see at all age group, the thinness was found to be all Himachali children, and the severely thinness was found only at the age group of 15. And this table shows none of the boys were found to be severely stunted. Conclusion: Himachali boys appear to be taller as compared to the most Indian population of boys of that age group. The prevalence of different categories of overnutrition and undernutrition was also lower as compared to the other Indian studies on adolescent boys. The existence of overweight and obese individual points towards the global burden of mind. Girls also selected for this purpose, but
why students from Himachal?
mostly belongs to I am only presenting paper.
Dr. Varsha, I'm also having 2 questions. Why only boys? Why not girls?
Girls and boys both are selected from the Himachal Pradesh.
Okay. And one thing which was required was the demographic data, socio-economic status of
Yes.
the families from which the students are.
Economic status
That
also
would be
included.
the deciding factor behind the reduction or stunting or that have been included in the study.
Yes.
And they are better because they have such a rich state having fruits and all. We all depend on Himachal and Jammu Kashmir for.
So for this purpose, non-hilly regions were selected.
Even availability of all the things is surplus over there in Mandi and Suttaragar. I have been there. I have seen that they are having a rich availability of everything there. As compared to some, as compared to villages of India, other villages of India.
Okay, thanks Dr. Varsha for your presentation. Now I request to OP28 for their presentation.
My research topic is CUB and Sushi.
Yeah, please fast.
So my research topic is the CUB and Sushi domain containing protein with assays-like activity confers insecticide resistance in Indian malaria vector and a phase 3 insect. So I work with malaria. So this is the life cycle. This is the life cycle of malarial parasite. So malarial parasite requires two hosts to complete its life cycle. One is the human and other is the mosquito. So mosquito transmits this disease from one human to another and acts as a vector. So one way to control this malarial disease is the vector control. And we have some vector control strategy. We can target the mosquito at the larval stage as well as at the adult stage. Whereas at the adult stage, only insecticides we can use in the form of sprays or insecticide treated nets. So we have limited class of insecticides. We have organophosphates, pyrethroids, organochlorines and carbonates. You must have known or heard about the DDT, which we were using earlier. And currently we are using organophosphate and pyrethroids. These are the latest class of insecticides which we are using. And the current latest class is pyrethroids, which were developed in the 90s. So we have limited class of insecticides and we are using them extensively, and this creates a problem of insecticide resistance in the mosquitoes. And this is the latest WHO's data. As you can see here, that in Indian scenario, India has reported resistance against all the 4 insecticide classes, and the situation is alarming, and this insecticide resistance mechanism is poorly understood in the mosquito, especially in the Indian mosquito, that is Anopheles stevensii, Indian malaria vector. So we intended to identify a new gene that we can correlate with the insecticide resistance mechanism. For that, we have taken a resistant and susceptible Anopheles stevensii strain which we have in our insect tree, and we performed SDS page and proteome analysis, and we have identified some differentially expressing proteins from the resistant mosquito and sent them for mass spectroscopy. And with this we have identified an uncharacterized and unknown protein from the mosquito, Anopheles stevensii. That protein is of 132 kilodalton size and contains 2 domains at the C-terminal region, that is CUV and Sushi domain. These domains, these proteins are already known for the drug resistance in cancer. So we studied this in the insecticide resistance for the mosquito. Next, this is the multiple sequence alignment showing that this protein is present in both the other Anopheles vectors. Next, we express this protein in the BL21 strain of E. coli, and we looked for its activity, and we found out that this protein has an esterase-like activity. So that we know that this protein, although this protein is not sequentially similar with the esterase, but can function as an esterase enzyme. So it can break the ester bond of the insecticide but we need to look whether this protein is interacting with the insecticide molecules or not. For that, we perform MD simulation, and that is an in silico analysis, bioinformatic analysis, and with that we come to know that this our protein is interacting with these insecticides. So we further validated this with the in vitro experiment that is surface plasma resonance, and we concluded that this yes, this CSDIR protein is interacting with this delta methylene and methylene, which are insecticides which we have used in our study. Then to functionally know the role of this protein in insecticide resistance, we perform RNA-mediated gene knockdown in the adult mosquito, and the gene knockdown was confirmed through the RT-PCR after 24 hours, and we achieved almost around 50% gene knockdown, and we performed this percentage survival assay to know whether this gene should be non-essential. That is, when a gene is knocked down, the organism must not be killed. So there is no effect on the survival. Then again, we performed the esterase activity assay, and the esterase activity got significantly reduced in the test samples, that mosquito test samples, and we can conclude that the esterase activity is of CSDIL protein only. Last, we performed the WHO tube adult susceptibility assay on the resistant mosquito, in which we have knocked down that gene, and you can see in the graph that the control has a less mortality, means they are resisting, and in the gene knockdown, the mortality got increased. The mosquito which were earlier resistant now becomes susceptible after knocking down of this gene. So we can conclude from here that this gene is somewhere involved in the insecticide resistance mechanism. So we can conclude from here that we have identified an uncharacterized protein with esterase-like activity from delta-methylene and methylene-resistant Anaphylis site. Our MD simulation and SPR data suggested a strong interaction of the identified protein with the delta-methylene and insecticide, methylene. And the gene knockdown data of CSDIR showed enhanced toxicity to delta-methylene and methylene in resistant Anaphylis site. Overall, this data suggested that the identified protein might be playing an important role in the insecticide resistance, and studying this protein further will overall aid the current vector control strategy. So, these are the information. I would like to and my lab and also ICBTM for giving us the opportunity to present our work here. Thank you. Fourth year.
So while using this insecticides, what precautions you took? You use this or you have done only computation?
No, we performed this.
Experiments.
Yes.
So these are very dangerous insecticides, and all these things. What precautions you have taken? Or you use it as such? No, these are highly.
No, like to expose the mosquito, there is a WHO paper, impregnated paper that comes from WHO. We use just normal gloves, and we take a tube and we put that paper inside and keep the mosquito for 1 hour and give the exposure.
Okay, but I just want to give you a suggestion that these insecticides, they are highly toxic.
Yes,
and they can cause toxicity in humans also.
Yeah. But we have used in very limited ways. Yes. For the SPRSA and this
We have some suggested model type oxide
tubes. Somewhere like a cylindrical tube, and we put a paper inside it,
Good morning everyone. Today me and my partner Javier will going to give a presentation on the topic comprehensive review on Alzheimer's disease. So before deep diving into the topic, I have two small questions from you guys, and the answer of first question lies in the second question, if you carefully focus on. So first question is obvious, which is very obvious. Why should we choose Alzheimer's disease? Like there are so many other options, so many more fatal diseases actually. So why should we focus on Alzheimer's only? The other question is, how many of you would want to forget or get forgotten by your loved ones? No one? That's the exact reason why Alzheimer's is important. So let me just present the epidemiological data in front of you guys, so that you can have a clear picture about the fatality of the disease and how enormous the disease is. So as you can see, first the definition of the Alzheimer's. Alzheimer's disease is cognitive dementia, which is a general term for brain-related issues, which comprises of combination affects the cognitive abilities and memory. The early symptoms of Alzheimer's basically express 10 to 15 years after the onset, which is very late, which is generally after the age of 60. So if we try to get a hold on the disease, it is actually already late. So worldwide, if you see the number of patients over the age of 60 which suffered from Alzheimer's was 897.14 million, out of which 46.78 million were affected by Alzheimer's. And this figure is expected to grow to 131.45 million by 2050. So if we focus on India only, then 7.4% of Indians above the age of 60 are living with dementia. So now I will hand over the further part of the presentation to Javi. Actually there are not specific mechanisms for why Alzheimer's actually happens, but there are four major hypotheses which are generally more focused upon and extensively researched upon. So the mechanism of the disease will be explained by Javier, then I will again take over the further part of the presentation.
Thank you, Rahul. Thank you, Rahul. Now we will have a look at the cholinergic hypothesis. As we can see in the figure, acetylcholine is a neurotransmitter that is used for the neurotransmission between the cholinergic neurons. So, according to this hypothesis, the cholinergic neurons degenerate, and that was the level of decrease of choline, acetylcholine. That causes the increase in acetylcholine esterase, which metabolizes the acetylcholine. This hypothesis states that due to the decrease of acetylcholine levels in the brain, that cholinergic neurons degenerate and hence causing the neuronal impairment. Next, we'll have a look at the amyloid hypothesis. The amyloid beta precursor protein is concentrated in the synapse of the neuron, and it acts as a cell surface receptor. In this hypothesis, it is usually broken down by gamma secretase and alpha secretase. When it is broken down by beta secretase, it leads to the formation of amyloid beta plaques. And I would like to tell that the plaques and tangles, as we will discuss in the next hypothesis, are the most common terms that are used while discussing Alzheimer's and the cause of Alzheimer's. Now we'll have a look at the next hypothesis, the tau hypothesis. The tau hypothesis states that as you can see that microtubule associated protein tau, MAPT, it stabilizes the microtubules through interaction with tubulin. But in this, when the patient is affected by Alzheimer's disease, it causes the hyperphosphorylation. You might think that why is it getting caused? It is caused due to the improper metabolism of glucose in the brain, and also caused by the improper cell functioning. This leads to the formation of neurofibrillary tangles, as I talked about the tangles that are in the brain, which leads to the improper functioning and the neuronal atrophy. Next, we'll have a look at the mitochondrial hypothesis. In this hypothesis, it states that the mitochondrial functions are decreased in the patients that are affected due to the Alzheimer's. As you can see in the figure, there is an increased level of reactive oxidative species (ROS), which leads to the oxidative stress, and hence it plays an important role in the onset of Alzheimer's disease.
Now we'll see some risk factors associated with Alzheimer's and the probable causes by why it happens. So, as explained by Javier, in the amyloid hypothesis, the obvious reason first is age, which is due to the cholinergic neurons degradation as the age progresses. The other reason is gender risk factor. So, it has been seen that female are more prone to Alzheimer's as compared to male. Now, earlier it was hypothesized that it was due to the higher longevity of the woman, but later it was concluded that it is due to the estrogen levels in the late females, which are very less. Now, if we see, then the younger females, which have more estrogen level as compared to the late females, are less prone to Alzheimer's. Now, we see the example, then a male of 60 years and female of 60 years, then the female of 60 years would be more prone to Alzheimer's. Now, next is diabetes, which Javier also explained in the tau hypothesis. The improper blood glucose level, brain glucose level metabolism is associated with the tau hyperphosphorylation, which is also the case in case of diabetes. So there is one correlation between Alzheimer's and the diabetes. Now mitochondrial dysfunction is already explained. Epigenetic factor, as we see, then the cell functioning modulation is controlled by gene expression is mainly done by non-coding RNAs, microRNAs, which are small non-coding RNAs. Regulation explained at post-transcriptional level in case of Alzheimer's and affect the amyloid beta processing. Now brain injury. Brain injury can cause severe damage to the blood-brain barrier, which may lead to the leakage of blood plasma in the brain, which can affect the patient. Now, vascular disease, there has been less data available that the researchers have found the correlation between the vascular diseases and Alzheimer's disease. Now, these are some FDA-approved drugs. These are some FDA-approved drugs which are targeted for treating Alzheimer's, although there has been no fixed drug that can completely cure the Alzheimer's, but the major drugs target only the symptoms of Alzheimer's. And these are some repurposed drugs. The data from the Wish disease, that has been these drugs has been repurposed. I can give you the data if you want. I have not included in this slide. So now let's see the current advancement in the field of Alzheimer's. So the current conventional treatments which have been followed in the research with respect to different hypotheses as explained. So
I think that's
Can I ask a question? The one thing which I would like to ask is related to the coding. What is the role of acetylcholine, availability of synaptic acetylcholine in memory? How acetylcholine is going to have a role in memory, recognition, memory, cognition?
Acetylcholine is basically a neurotransmitter which helps in upregulating the cognitive functioning.
How?
Not the exact reason.
Actually, it helps in the neurotransmission of the signals between the cholinergic neurons. and in Alzheimer's it is decreased, as we discussed in the slide. The exact mechanism is not yet studied. Basically, it is the level of acetylcholine that regulates the pathway.
And what about the neurodegeneration? And certain drugs, levetiracetam, piracetam, you haven't mentioned that. The drugs which are very commonly used these days. Have you heard of piracetam, levetiracetam?
Hello.
You discussed four hypotheses. As per your review, which hypothesis would be the best for them?
The best would be the mitochondrial hypothesis. Because it is interlinked with reactive oxygen species, which is like the main concern because it interconnects with other reasons as well. Like in case of females, they are more prone. As I said, they are more prone to Alzheimer's. So it has been found that there are large number of radical oxygen species that has been found. So I think the mitochondrial hypothesis will be the more targeted one.
Thanks so much.
Next, please.
Thanks, Rahul and David, for your presentation. Now I invite OP30 for your presentation.
Very good morning to all of you. I am working as ICM RSI under the supervision of Dr. Shri Mishra from CSI CDRI Lucknow. So I am here to present my research work, which is titled Elucidating the Role of Base Excision enzymes in the Alveolar Genome Maintenance. So here I start with the introduction. So malaria is a mosquito-borne disease and which is caused by a plasmodium parasite. And as we all know about the malaria life cycle, so here the plasmodium undergoes the various stages of parasite life cycle where application takes place. So in all of these stages, lots of meiosis and mitotic divisions what happens. So during the replication process, the endogenous and exogenous DNA damages are inescapable. So all cell requires some form of DNA repair pathway for the normal product of their proliferation process. So here we target the base excision repair pathway, which is least targeted and least studied and more conserved pathway in all of the Plasmodium species. So here lots of enzyme works in the base excision repair pathway. So my work is focused on glycosylase, API aminonuclease and axonuclease pathway and DNA ligase as well as. But due to the time limitation, I only explain the glycosylase enzyme and API aminonuclease enzyme. So these are my results. OGG1 and AP1 are dispensed during asexual blood stage propagation. So here we demonstrate the role of OGG1 and AP1. OGG1 is a glycosylase enzyme and AP1 is the endonuclease. So to demonstrate the role of these enzymes, we do the phenotypic characterization of these proteins to develop the knockout parasite of these enzymes using the double crossover homologous recombination strategy. And the knockout parasite was confirmed with the GFP expression which inherited in the knockout parasite, and as stage for cassette for the knockout parasite development. So the GFP expression confirms the knockout parasite, and these are the diagnostic PCR which further confirmation of the knockout parasite. To restore the function of original gene, we do the complementation study and also restore the function of these gene and confirm by the diagnostic PCR. To check the role of the OGG1 and AP1 in the blood stage, we counted the blood growth curve, but using the 0.5% blood parasitemia, and there was no significant difference in the blood stage in comparison to the wild type. So OGG1 and AP1 are just dispensable for the asexual blood stages. Now we check the role of OGG1 and AP1 in the parasite development in the mosquito stages. And for the mosquito stage development, we fed the mosquito with the infected knockout parasite mice or also with the wild type for the comparison study. So the development of the oocyst inside the midgut, the development is completely normal in the case of both knockout parasite in comparison to the wild type, and also counted the number of oocysts in both of the knockout parasite. And on day 18, we check the number of sporozoites inside the salivary gland because it's transmitted to the salivary from the midgut. The number of sporozoites is also normal. You can see that the lobes were filled with the GFP expression parasite in the knockout in comparison to the wild type. So these proteins are also normal for the mosquito stage development. If we further moving on to the liver stage development and liver to blood stage transmission to the knockout parasite in comparison to the wild type. For check out the infectivity of the knockout sporozoites in C57BL6 mice in comparison to the wild type, and we found approximately 2 to 3 days delay in both of the knockout in comparison to the wild type, because the wild type is positive on day 3, but in the case of knockout, it approximately 2 days delay. We repeated the experiment in the 3 biological replicates, and we found same delay in the knockout parasite. And also we counted the number of day 7, counted the number of parasite on day 7. In both of the case of knockouts, we were found approximately delayed appearance in the IRBC, infected RBC, in case of knockout in comparison to the wild type. So to find out the reason behind this delay, we further check out the development of the knockout parasite development in comparison to the wild type in the in vitro condition. So OGG1 and AP1 knockout parasite, for the in vitro experiment, we infected the HG2 cells with the 5,000 sporozoites of knockout, both of the knockout sporozoites and also with the wild type for the comparison study. So this is the early stage development of the liver stage, and this is the late liver stage development of the parasite. And we didn't find any kind of visual defect inside the liver stage development, and which is further confirmed by this statistical data. So the number of EF and the nuclei count and the EF areas are completely normal. But the reason of this delay, due to the when we do the in vitro experiment, the mirosome development was not efficient to stable increase the infection inside the blood stage transition. When the mirosome develops, we inject it inside the mice and we check the infectivity of the mirosomes of knockout sporozoites in comparison to wild type. So the mirosome was unable to initiate infection in the blood stage when it passes to the liver stage. So another study of mine is AP1 double knockout development of the parasite. So one more homolog present in the plasmodium is AP1, and AP1 is I already explained this role. So maybe these proteins are redundant in the plasmodium genome. So we check the cumulative effect of these proteins inside the asexual blood stage development of the parasite. So we developed a double knockout of the parasite, in the plasmodium genome. We transfected the AP1 construct which expresses GFP expression inside the PB AP1 knockout mCherry expressing cassette which having mCherry expression. So after the transfection process, using this reverse genetic approaches, after the transfection process we generate the knockout parasite. But when we further doing the WR drug selection, so under the WR drug selection, only HDHFR and YFCU having cassette was survived, which is double knockout parasite cassette. So after the WR drug selection, we don't find any kind of parasite in the case of double knockout, which confirms that when we deleted both of the genes simultaneously in the one genome, so these gene is essential during the asexual blood stage development. journal of DNA repair, and are already doing some other publication, but these two projects are my under revision. Thank you.
House is open to the questions. If anyone has any questions? All right.
Thank you, Himanti, for a good presentation.
I just want to ask, these are your blood state भी किया है, इसका liver state भी किया है?
Actually, this is firstly we check on the blood status when it survives. So we further got some parasite in the blood state, then we next tested in other stages, but we didn't found any kind of parasite in the blood state. We're not doing it, we just find out that it's a novel drug target. So further we develop a compound and further we do the clinical trials and all. It's a future study actually.
Now I request to OP 31 for your presentation.
Transmission and mosquito development. So, going to the brief introduction, we know that vector-borne disease accounts for more than 17% of all the infectious disease and includes around 7 lakh deaths annually. Out of which, malaria itself includes the 4 lakh deaths per year and account for 219 million cases globally. This malaria is caused by Plasmodium parasite, which is transmitted to humans via Anopheline vector. And these Anopheline contain a lot of species which able to transmit this malaria. However, these species are confined to the continental boundaries. Like in India, we found Anopheles stephensi and Anopheles felicis, while in America, we find this Anopheles darlingae, Anopheles gambling. So, what causes these species to be in the continental boundary? But in the recent years, it has been shown that there is an expansion of Anopheles stephensi from Asian subcontinent to the African subcontinent. It means they are adapting different environmental conditions also. So when we go to the literature, we find that heat shock proteins are crucial for adaptability of Anopheles. So there is a several stress-like conditions like temperature stress, parasitic stress, or the insecticidal stress, in which the heat shock proteins are responsible for adapting to the new environmental conditions, which could be a good target for vector control strategies and to inhibit the parasite transmission in the, to the human population. So what are HSPs? Heat shock proteins are the molecular chaperones that are conserved in all the species and maintain the protein quality in a cell. It means they maintain the hemostasis. Whenever there is a stress-like situation, they fold the unfolded protein and denatured protein and activate that protein. So there are different classes of heat shock proteins like small HSPs, HSP 27, 40, 60, 70, 90, and being named on the basis of their molecular weight. HSP 70 and 90 are the crucial proteins, but my work is on HSP 70, so I will elaborate about HSP 70 here. HSP 70 includes a two-domain, nucleotide binding domain and a substrate binding domain. I will talk later about the mechanism. First I will show you the results. So what we did, we prepared a catalog in Anopheles stephensi and Anopheles felicis, which is an Indian malarial vectors that is responsible for transmission of malaria. We find 7 HSPs in stephensi felicis and we compared these HSPs with the global Anophele gamete. So we expressed in the different stages of mosquito cycle, but they are mainly regulating during the larval development. From this, we take the random hit of one HSP1 of the anopheles. Coming to the mechanism, this HSP70 requires HSP40 for its working mechanism. This binds to the unfolded protein, and in the presence of ATP, this protein gets folded. So we express both the HSP70 and HSP40 to evaluate its enzymatic activity. We find there is a minor difference between the activities in presence of HSP40 and in absence of HSP40. Next, from the literature, we took two compounds which already known to bind HSP70 or already known to inhibit the expression of HSP70. We did the surface plasmon resonance to calculate the binding kinetics of these two compounds with our protein. So we found that this compound 1 has a binding constant around 300 micromole and the second one is around 200 micromole. We then tested these two compounds for the larval development, whether these are able to inhibit the larval development or not. So we first tried on eggs. We treated the eggs with these two compounds and observed for 72 hours. We find that compound 2 is inhibiting around, showing the IC50 around 12.5 micromole. And we next treated the larva with these two compounds. We find, like saying this compound 2 is working best. We then treated with pupa also, but there is no significant difference in the emergence of pupa to adults. Because pupa is a non-feeding stage, the mosquitoes are not getting these inhibitors. Next, to show the role of HSP in sporogonic development. So what happens? This malaria parasite has a two hosts, the vector anopheles and the human host. The sexual development of this parasite occurred in the anopheles vector. So we targeted the three stages of this plasmodium parasite: exflagellation, eukinete, and the oocyst stage. And from the literature, we have gathered the information that this HSP72 is highly regulating during the plasmodium falciparum infection after the 7th day. And this 7th day is for the oocyst day. Like this takes around 1 to 2 day information, then it forms the eukinete, and then after the 7 days, the oocysts were formed. So we find that this compound 2 has a no effect on both exflagellation and eukinetes, but during the 7th day, the number of oocysts being reduced by 90%. As you can see in the figure, there is a less oocysts up to the 10 micromole. From this, we have concluded that HSP70 emerges as a pivotal player for the mosquito development as well as for the adaptation to the microenvironment, and this HSP70 can be a good target to reduce the malaria burden because it also reduces the transmission. So I acknowledge my supervisor, Dr. Kailash Pandey, and my co-supervisor, Dr. Soumyananda Chakraborty, for helping me in this project, and my collaborators, funding organizers of this conference, and my funding authorities. So thank you, and if anybody has a question.
Any questions from audience?
Now, thank you, Bharti. Now I request to 33 for your presentation. 33.
from Humayun University, and the title of my today's talk is Molecular Assessment of Biodiversity of Himalayan Flora and Bioprospecting for Anti-Cancer Regimes. So this university is located in the mountainous state of Himalaya, and you know that Uttarakhand is also known as the Habal state. Because 45.44% of the geographical area is covered with forest, plus there is a lot of topological variations, temperature variations, different terrains, so that leads to biodiversity rich state. Okay, so the rich biodiversity must have been, can be expected to be a boon for any state in terms of environment as well as in terms of say bioprospection and economy. But this domain of biodiversity is currently facing a number of challenges, like the overexploitation of bioresources that is driven by corporates or the local players. And there are, even if something is confiscated, you cannot, and if it is in the pulverized form, you cannot basically confirm the identity of that thing, the plant material. There are many cryptic species also, like in lichens, in fungi, there are many cryptic species which are very difficult to identify through conventional means. So, and we know that, the identification is the first step towards conservation. Until we identify the species, we cannot conserve them. So, particularly that happens in lichens, okay? And in addition to the rich biodiversity, because there are many tribes which are living in the state and which are segregated from each other by geographical means, and because so they are having their own pharmacognosy. And the thing is, like the medicines used by them is very effective, but they have not been appreciated by the scientific community because they are not scientifically validated. The second thing is, so what happens? There is no prior art, right? So what happens? Like the companies which are overexploiting that biodiversity, they actually they basically steal that conventional knowledge, and they make their drugs, and they never give any credit to the local community or financial thing to the local community. So one thing is biopiracy real? The reason is we are not getting any news, right? We don't have any news. We don't see very often these kind of news that okay, this is biopirated. But the thing is, it is an ongoing process. So no news. In case of biopiracy, no news is not a good news, right? So what is the solution? Solution is, what we can do is we can identify molecular markers, metabolic signatures, okay, like the DNA barcodes, and associate them, document them with the traditional knowledge. Okay, that is one part, and then we can validate the traditional medicinal knowledge with the modern scientific approach. So that can possibly address all of these issues, like the cryptic-like forms you can identify through DNA barcoding, like say unavailability of prior art. So you are having your data, like the bioprospection data, so that can be addressed. So on this line, what we did, we actually created we collected more than 350 lichen species belonging to more than 60 genera from Uttarakhand, Himalaya. We carried out their DNA barcoding. We generated PLC fingerprints for them. I'll show some data, some representative data, because that's a huge amount of data. And we have made high resolution databases of images of these lichens. Okay, and along with them we have also barcoded mushrooms, interlichenic fungi, bryophytes, and high-value plants under different projects. And we are also carrying out their bioprospection as well. So, like these are the few lichens. So we have developed so many, more than 700 pics, high resolution pics of these lichens of Uttarakhand, Himalaya. We have more than 350, pics, these kind of pics. Like this is developed by SQS4. This is through UV, low wavelength UV, and this high wavelength UV. And we have done DNA barcoding for more than 60 species. Additionally, we have done spot tests, that the keynote taxonomy part, for more than 350 specimens, and bioactivity assessment for approximately 10 species, and GCMS of lichen metabolites for more than 20 species. So we also, as I told, we also done DNA barcoding for more than 40 high-value plants, Himalayan plants, under our DRDO project. These are a few of them. And we are then in the bioprospection part, we are trying to see their anti-cancer activity. Why anti-cancer activity? Because we know that many of these plants or lichens, they have been used as anti-inflammatory drugs or medicines in the traditional medicine. So we know that the inflammation is associated with proliferation, cell proliferation, as well as with the EMD. Here I will be studying. We will be studying. I will be showing the data for data for this thing. Proliferation inhibition as well as for the EMD. EMD is epithelial to mesenchymal transition after once the tumor is formed. Now the next step is metastasis. So in this one, what we do is we will be studying EMD as well as the proliferation part. So we know that free radicals are important and anti-oxidant activity is also important. So we have tried to see the phenolic content, flavonoid content. We can see that the flavonoid contents are really high in lichens, whereas like these ones are the plants we have tested. And overall phenolics are they are in higher amount in this thing. And this is a heat map, so you can see that okay, different plants are having different levels of antioxidant activities. We have tried to see the DNA damage prevention activity because of lack of time. I won't be going to the mechanism, but yeah, this is a representative figure, and we can by putting extracts, different extracts in a dose-dependent way, we can see that how much protection they are giving in a biochemical assay. And we know that the biochemical assay cannot be the full-fledged, cannot be conclusive. So we are also planning to do the gamma S2X immunofluorescence-based in vitro assays. Then, anti-inflammatory activity, we used these THP1 monocytes and then we converted them into macrophages through KMA treatment. And after that, we induced them to through LPS, and then we check the efficacy of extracts in inhibiting the LPS-induced inflammation. So they are not heat map, they are basically bar diagrams. So this is control, this is lower dose, and this is higher dose. This is for, we check TNF alpha, TGF beta and IL-1 beta because they are inflammatory markers. Then we also examine their acidity values, you can see here in different cell lines. So this one for the U87LG cells and this for the N67 cell. And we also examine the same thing through the expression of proliferative marker like Ki67 and BCNA. And then the wound healing assay, because wound healing tells you about how much your extract can inhibit the migration of the cells. And along with that, we have also examined the expression of mesenchymal markers like the WIM, JAP and TGF beta. So this is the conclusion. Okay, development and documentation of DNA barcodes and metabolic signatures is crucial. Okay, and we know that many of the studies have not been done at the protein level. There are scarcity of resources, but we are working on them. And we understand that bioactivity guided purification would be useful. So thank you so much for your attention. Yes.
I would like to know what was the basis behind the selection of plants, and secondly, what was the basis behind selecting those plants for anti-cancer?
Yes. So actually, for the plants, these plants were given to us from the DRDO. Okay, they wanted us to work on them, their bioactivity and their barcodes. And like in lichens, because we are lichen barcoding has not been done earlier. We have done just around 60 or 70 barcodes, but still we are means our lab is having maximum number of DNA barcodes nationwide. So very less work has been done. So we have we selected them randomly, and we took clues from traditional knowledge, like whatever is available, for examining their this bioprospective potential.
Traditionally, they may not be using these plants for cancer because cancer was not.
Yes, ma'am, that's what I told. Actually, yes, ma'am, you're right. Because they are using these plants and lichens as anti-inflammatory agents, and inflammation is something that is inducing proliferation as well as EMD. So that way, because they
Innovation is related with so many diseases.
yes ma'am.
Why only switch towards the cancer?
Yes,
There
ma'am.
may be diabetes, there may be some other conditions.
Right, ma'am.
Metabolic disorders.
Well, we have explored diabetes also, and we have explored the because traditional knowledge is available for the antimicrobial activity also. So antifungal, antibacterial activity also we have done, but I have not shown yet. Yes.
You have done the activities on illness. What have you concluded? There are studies going on.
Yes, ma'am. The study is going on. That is one part, and yes, ma'am. And the second part is, we have done multiple assays, additional assays for individual plants, but because they have not been done across the board, so that data couldn't find a space error. Yeah. Thank you so much.
Now I'm going to invite to OP34. OP34 for presentation.
I request participants to take care of the time limit as we are already running short. So after the prize, please, I would request you to stop very because time is where we are really very long.
Good afternoon, all the panel members and all the audience. So, I am Nirdosh, UGC Jara, under the Dr. Satish Mishra from CSIR CTRI. So, my title is an immunoinformatic approach designs a multi-epitope vaccine candidate for Plasmodium secretory proteins. So, please play the video. As you know that the malaria parasite has a complex life cycle and it starts when the female Anopheles mosquito bites a healthy individual. There, these lively gland sporozoites release a number of proteins which either modulate the host immune response or they start the initiation of the pathogenesis and then travels to the hepatic circulation. So these are the two steps which we targeted for our vaccine construct. And one data which we found from the Advil et al. That there are 477 proteins which are specific to the slimy gland sporozoite cyst, and here you can see the video. Please play. During the slimy gland transmission, these sporozoites are traveling to the skin to the from the female Anopheles bite, and they are secreting a number of proteins. So I started the proteome retrieval from the Plasmodium database, and I found that whether these proteins have secretory in nature or not. So out of these 477 proteins, 91 proteins having this signal for the secretory in nature. For any vaccine construct, we need a protein which is antigenic in nature and non-allergic. For that, we excluded or filtered out these proteins and found that there are 9 proteins which are secretory in nature and 3 invasion hepatocyte invasion proteins like celtose, STRAP and EMP3. And further we evaluated and demonstrated the epitopes. So we used the MSC1, MSC2 and B cell epitopes, including the T helper cells and T cytotoxic cells. And then we took all the epitopes which having the overlap for the B cells, T cells and T cytotoxic cells, and then we constructed a primary linear structure of the protein with the adjuvant which is already adjuvant, that is HPS-AG in the RTS malaria moxirix vaccine and in the R21 which is recently imposed by the WHO. So we made the secondary structure and then the tertiary structure, then refinement, and we did the docking with the TLR. So this British portion of this cartoon is suggesting representing the adjuvant, that is HPS-AG hepatitis surface antigen protein, and this greenish portion of the cartoon is representing the epitopes, which we found out fine. And these whole construct of the vaccine protein is it's covering the, we checked their population coverage for the geographical, racial and coverage, and we found that there is 86% population will be covered by this vaccine in comparison to the right now vaccine currently available R21 and Moxex, which is having 26% population coverage. These are the allelic table. I'm sorry, it is not visible here. So these are the vaccine should be like these vaccine constructs should be in the surface should be in a close proximity so that they can bind with the B cells and T cells. And here the epitopes with the these sequences representing the B cells binding binding site in the yellow portion. So we got 14 epitopes where B cell, T cell and T cytotoxic cells will bind, and further check for the immune stimulation whether these protein vaccine construct is eliciting the immune response in the body or not. So we found that these graphs suggesting that the vaccines should be eliminated within 5 days, and the IgM IgG complex will stay longer for beyond 35 days, and same with the intelligence, TNF gamma and other immune responses we are getting here. So these are the immune simulation results which are depicting that these vaccine constructs will elicit the immune response with the B cells population and titer, and T cell, cytotoxic cells, and the overall titer and the concentration of the B cell, T cells remains constant beyond the 30 days. So there is a paper, Jiaozu et al. So they suggested, they found that in the biological system, the parasite is more preferentially binding with the TLR4 rather than TLR2. So just to check that whether our construct is depicting the same thing or not, we did the docking study with the TLR4, these TLR4 in the greenish cartoon structure, and this is the protein construct. So for the binding affinity, we found that it is showing that minus 1235, and in comparison to the TLR2, which is having minus 1031. So this result suggests that the vaccine construct is same with the biological data, it's more preferentially binding with the TLR4, which also validate our study. And then for their complex the stability, we did the molecular simulation and the simulation, and we found that these protein complexes, both the protein complexes are in a close proximity and in a complex form, and they are stable for the 15 nanoseconds. So in conclusion, I will say that these vaccine construct is more have potential, and we are planning to do the biological experiments in mice, rats, and rabbits. So we are thankful to the CSI and CDRI and UGC for the fellowship. These are the references.
Any questions?
So you are from where?
CSI and CDRI.
PhD student?
Yeah.
So you have done computational work? All computational?
Yes, ma'am. All right. But ma'am, last study, which we did the document study with the DLR, which is replicating the biological system. That there is Jiaozu et al. The published paper in 2023, and they said that the parasite, parabolic parasite, is more preferentially binding with the DLR4 rather than DLR2, and our docking study and computational data suggest the same thing, and it replicates the results.
So you are doing PhD with whom?
Dr. Satish Mishra. कहाँ से हैं आप? CSIS 11
First
CBA.
line first line.
Final year student या first year?
Second year. Oh, nice.
Okay, thank you very much. Now I request OP22 for a presentation.
अभी कितने रह गए हैं?
College, University of Delhi. Today I will be presenting my work. The topic of my work is metal-free construction of aminated isoquinoline frameworks to observe two alkyl benzohydride in an aqueous medium. So in this work, what we have done, we have used a metal-free protocol for the synthesis of various aminated isoquinolines in an aqueous medium just by using water as a solvent. The reaction time was very less, which is 3 to 4 hours. So, before talking about my synthetic protocol, let me tell you something about amino isoquinolines. They are well-known bioactive molecules, which are used as antituber agents and also in anti-Parkinson's disease. So, these are all the previous quoted protocols for the synthesis of aminated isoquinolines. They have used various metal-based synthetic protocols and a very tedious protocol like reaction in toluene as a solvent using 8 hours, 110 degrees Celsius. Also using palladium catalyzed reactions. But in our work, what we have done, we have optimized the reaction conditions under water as a solvent in 4 hours, and the yield was also promising, which is 99% of yield. So we have used this starting material and various amines. We have used secondary amine, primary amine, some drug-based amines also for the synthesis of diversified amino isoquinolines. These are all the substrate scopes these are all the compounds that we have synthesized. We have synthesized there about 50 molecules using this scheme. These are from the primary, these are from the secondary amine series, and these are from primary amine, and these are also from primary amine series. Also, we have used this secondary amine as well as primary amine based amine. And both the reaction on both the nucleophilic centers, the reaction underwent very well, giving a yield of near about 85, more than 85% yield. These are some of the drug molecules that we have incorporated in our schemes. These are used in anti-Parkinson agent, influenza, and also used in viral fevers like this one. And this is the gram scale synthesis. We have used 1 gram of starting material, and we have gotten 1.8 gram of final product. These are some of the mechanistic experiments that we have performed to check the mechanism of our protocol, of our synthetic protocol. These are all the experiments that we have done, and also we have checked the deuterated proton that is coming in our product. It is coming from the solvent. Like if we use
use H2O, it will be H, and if we will use D2 as a solvent, it is converted into D. So this is the mechanism. This is the nucleophilic addition and then subsequent annihilation to get the desired product and the yield 98% yield. So this is the conclusion. We have used a metal-free activation of nitrites in aqueous medium. We have synthesized diversified comminuted acetylene using a very economical and clean methodology. All the functional groups reacted very well under this scheme, like electron-rich, electron-deficient, various amines underwent the reaction very well. This work has been published in Journal Review Chemistry. So lastly, I would like to thank my lab members and my guide, Dr. Dinesh Sharkey, Dr. Priyamada ma'am, and also everyone sitting here and listening. Thank you.
What's your name?
Imanshi.
Imanshi, I have one question. You have performed the reaction with aqueous conditions. So, have you used no catalyst? This completely propagated
Yes, ma'am, complete water. No catalyst was required.
Have you optimized these reactions in other solvents and
Yes,
solvent free?
Yes, ma'am. In DMSO, DMF, every solvent that is available.
And solvent free?
Yes, ma'am, solvent free also, but the yield was not promising.
Okay, thanks so much for the nice presentation. And now I request OP23 for a presentation.
I'm here to present my work. I'm actually working on phytochemicals as a potential strategy to treat breast cancer. So, I'll briefly introduce why breast cancer. So, we all know breast cancers in all are very most common disease that are causing most of the mortality across the world. Coming to breast cancer is the second most frequently diagnosed cancer in women across the world. And recently, last year only, India became the first country to have the most cases of breast cancer in women and as well in men, women and men both. So, if I talk about the statistics of the state-wise breast cancer data in India, the most breast cancer cases are found in Kerala, that has 45.7% of the breast cancer cases, followed by Punjab, that is 43.4% cases. Also, the main causes and symptoms that include, of course, the DNA mutations and specific disablement of various functions. The symptoms and causes are aggravated by age, risk factors like age, hereditary, lifestyle, sedentary lifestyle, basically smoking, alcoholism, and hormone, of course, hormone imbalance, that is progesterone, estrogen, or other HER receptors as well. And also diabetes and obesity. These are the major symptoms, risk factors that cause the aggravation of symptoms that are lumps in the breast cancer, breast or nipple pain, change in breast color, nipple discharge, skin irritation, armpit pain, etc. These are the basic symptoms and risks that are included in breast cancer. Okay, the current treatment strategies involves mainly surgery, radiotherapy, chemotherapy, also HER2 targeted therapy, hormonal therapies. That the course of the treatment strategies depends on the type of or the stage of the breast cancer, but there are limitations that are common to all the strategies, that is drug resistance development, effects are loss of hair, fatigue, vomiting, pain, nausea. That are common for all other cancers as well. So what are the alternative strategies that we can use despite of these? So there comes the role of phytochemicals. Phytochemicals are basically bioactive natural substances that are derived from plant parts. They can be root, leaves, seeds, or leaves and flowers. There are over 5,000 types of phytochemicals present in tea, ginger, pomegranate, all the foods generally we consume in India. The mechanisms that these phytochemicals target are the induction of apoptosis, that is the killing of the cancer cells. Okay, cell proliferation inhibiting the growth of the tumor, anti-inflammatory differences, anti-angiogenesis effects. I'm sorry about the clarity of this slide, but this was a beautiful diagram I draw. I'm sorry. Okay, so all other phytochemicals target these mechanisms, and the phytochemicals that are mainly very popular in India in our spices and common fruits and consumables that we use are in our regular diets are curcumin. That's from Okay. So next, what we did in this work was the in silico screening of phytochemicals and of course some synthetic compounds that were synthesized in my lab by my colleagues against the phytochemical metabolic enzyme, that is LDHA, lactate dehydrogenase A. It is a enzyme of the metabolic pathways which is generally rewired or reprogrammed by the cancer cells. It is mainly used in the interconversion of pyruvate and lactate with the interconversion of NADH and NAD. These are the cofactors that are necessary for the working of this particular enzyme. The role of this particular enzyme is in the development of cancer, tumors, invasion and metastasis. Ubiquitously, it is present in all the tissues, but it is enhanced in cancer cells. Okay, it also serves as an important checkpoint in gluconeogenesis and DNA metabolism. Why it is important? Because it is enhancing the angiogenesis processes of the cancer cells, and it also prevents the apoptosis of normal cells, preventing them converting them to the cancerous tumors. Okay. So in this study, we prepared a library of 252 phytochemicals that was available on PubChem. We screened them against LDHA, against the proteins 5WK, 5WKJ, and with and without cofactors, because the cofactor NADH has a role in the working of this particular enzyme. So another library was for 355 synthesized compounds that are all novel compounds. Among these, we got hit compounds after site-specific docking, that is particularly for LDHA target only. To further verify our results, not verify, actually validate our results, and to come to a shorter version of compounds, we did the off-site or blind docking to check whether these compounds do not bind to another target or site in the body against other than the LDHA. So those results that was promising was the most one was the hydrazine and catching gallet. And among the synthesized compound we got 6172 and 6178. We also performed the off-target study and LDHA sequencing of LDHA particularly enzyme, and we found that among the 78 most similar sequences, the LDHA family of LDHA actually has many different types of isotypes. So LDHA was most similar, and it was all similar to the human organ, no other forms of targets of the enzymes. Okay, this is the result. The binding affinity of hypercine was 9.2 minus 9.2 kilocalories, and 6178, that is the most promising in this study, was minus 9.0 kilocalories. We also performed the molecular dynamic studies for like 150 nanoseconds. Of then the compounds, this is hypercine, the most phytochemical, and it was stable after 60 seconds. That was our control that was already embedded in the protein structure we took. And this is the synthetic compound that we took. And it is all these the ligand and protein interaction of all these three compounds are very promising. So we have taken this to the wet lab data validation. I'm currently performing the wet lab data screening of these compounds against breast cancer cell lines. One of the data is already available for hypercine, but I'm not sure because it needs to be repeated. This is the future
I need to perform the secondary assays involving in vivo analysis of the data. And to conclude my study, I can say in silico study suggests hypericin as a phytochemical is a potential anti-cancer agent, and we can use it to the synthesize novel 6178 compound, and then we can probably get a better result for lesser toxic synthetic compounds, or even a completely phytochemical without any toxicity. Finally, I would like to acknowledge Dr. Brijesh Rathi, my co-supervisor, Dr. Geeta Singh, my lab mates, DRDO, and other agencies for funding it.
Nice presentation. Just forward your name.
Monica Kumari.
I just want to ask, you have mentioned it 355 human compounds.
Yeah. Ma'am, actually, our lab is an amalgamation of biologists and chemists. So my chemical counterparts already presented their work. So this is a whole library of chemical compounds that we synthesize in lab.
351 compounds is in your laboratory.
Yeah.
You have
Data for all.
And you have performed docking on all these things.
Yeah. Yes. Thank you.
Today is the last presentation of the 27.
My name is Dharmin. I am PhD student. PhD student from Uganda Hospital under the supervision of Dr. Prema. So my previous topic is antimony evaluation of compound isolated from the structure kinesis plant extract, which is the common name is snake meat. As we know, malaria is caused by the Plasmodium protozoa, which is transmitted by by the bite of infected female Anopheles mosquito. There is 5 types of Plasmodium which are affecting the humans. Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malaria, and Plasmodium lovelsi. Plasmodium is the main common one is the Plasmodium falciparum. According to World Malaria Report, about 247 million cases of malaria was reported out of 61,000 deaths were reported, which was very too high. The drugs which are being used for anti-malaria, chloroquine, artemisinin, meperidine, primethrin, or permethamine, and curare. And there was a first vaccine at 2009 for RTS-S. Although the first malaria vaccine it is not so efficient because it requires multiple boosters, and the drugs we are using have multiple resistance, so we need a drug which have more potency. That's why we have moved toward the new drug discovery. In my work, I have just isolated and verified, characterized the compound from isolated from the plant Streptococcus kinesis, and their biological study is done. There is a literature survey why we have chosen this compound, because this compound's ethanolic extract is being tested on anti-malaria and against leishmaniasis, because leishmaniasis and anti-malaria both are antiparasitic disease, and why we have what is the advantage of natural products? They are biodegradable, they are less toxic as compared to the synthetic one. They have health benefits. Their renewable can be regenerated. Work plan. This is my work plan. I have just reflux my plant extract with the organic solvent and purify with column chromatography and analyze by TLC and then characterization. And how this work is done? Refluxing with ethanol, filtration and chromatography, and there is a pure product. And this product is being characterized by HNMR. This is the final product. This is carbon 13 and HRMS. The peak 429 confirm the formation of this compound. And I have the data of X-ray crystallography data, which is not being included in this slide. And this is the biological data. The IC50 of this compound is being tested in antimony while 0.4 micromolar, and this is the hemolysis curve, which confirm that this is not toxic. Conclusion: the compound was isolated from the extract using chromatography and characterized by spectroscopy, HRMS, C13 HRMS. In vitro studies is performed against PF3D7, and further intermediate assays being in the study for the potential compound. So I would like to thank my PA, Dr. Premada Singh from Canada House, and Dr. Vijay Starkey, and all my class friends.
What kind of standards? What standard could be used
Now I hand over the mic to the Union for your further proceedings.
Thank you, sir. Thank you all the participants for exploring the intersection of science and care. I am glad and my pleasure to assist you. This was a great experience to everyone. Ending of this technical session, I would like to invite Dr. P.K. Chaudhary sir, please come and give the certificate to Dr. Komal Sharma ma'am. Sir, please. Thank you, sir and ma'am. Now again I would like to invite Dr. P.K. Chaudhary sir to give certificate to Dr. Shikha Agarwal ma'am. Thank you, sir. Now I would like to invite Dr. R.C. Amita sir to give certificate to Dr. Priyamvada ma'am.
Thank you very much, madam.
Sharma ma'am. Facilitation form. Thank you, sir. Now, I would like to invite Mr. Mr. Dinesh sir. Please give this felicitation certificate to Dr. Rajesh Aduria sir. Thank you so much, sir. On the behalf of ICPTM 3.0, I, Dr. Gunjan Javon, Professor, Pacific College of Pharmacy, Udaipur, to thank you for active participation and insightful contribution throughout this technical session. I want to express my gratitude to each of you and your engagement and dedication to achieving this research and practice. Thank you so much. Safe travel and until we meet you again. Thank you all.